As the small rosettes come up in spring, it's easy to pull them. You'll sometimes get an irritating tiny spine that festers under your skin for a couple of days. It never quite feels like you've gotten the full root. Your efforts, in fact, were in vain, as soon more and more rosettes just pop up anyways. By midsummer, there is an endless sea of thistle plants, and the fine hairs from seeding can almost look like snow on the summer breeze. Today we will be discussing the perennial colony-forming invasive weed Canada thistle. I'm Christy Gladham and I've worked in biological control for over 10 years. Welcome to the Colorado Department of Agriculture's Biological Control Series in collaboration with the USDA Western Sustainable Agricultural Research and Education Program. First introduced to North America in the 1600s, Cirsium arvense is a colony-forming perennial thistle species that may be called Canada thistle, California thistle, or creeping thistle. Canada thistle may be the most commonly used name in the western U.S. Despite these names, Cirsium arvense is native to Eurasia, where it can also be considered a pesky plant. I usually find that thistles are the most misidentified weeds in the field, so please take time and careful consideration to properly identify your thistle. There are at least 20 native thistle species in Colorado and over 80 native thistle species in North America, some of which are threatened, endangered, or limited to very small geographic regions. All of these native thistles are important parts of the ecosystem and our natural heritage. There are roughly 5 invasive non-native thistles in Colorado. Canada thistle may be differentiated from other thistle species in a few ways. The purple, lavender, sometimes white flowers are usually in clusters, not single flowers. They are also smaller than most other thistle flowers, only around 1.5 centimeters or about the size of a shelled peanut. Canada thistle does not have spines or wings on its stems. The stems are typically fairly smooth and round. These plants form large colonies that persist from year to year and typically continue to expand. If you start to dig one plant up, you'll find it's attached to an extensive fleshy root system with lots of horizontal underground growth. Keep all of these properties in mind as you assess your thistle infestation. Biological control agents are extremely specific to their target host species and will not be able to feed, reproduce on, or control a different species of thistle. If you are unable to definitively identify a thistle species, take photos and reach out to local native plant groups, extension agents, or other knowledgeable professionals. Canada thistle will begin growing in spring, sending up small rosettes when average weekly temperatures are around 41 degrees Fahrenheit. These rosettes are susceptible to competition from faster growing plants and are not very shade tolerant. Leaves are typically between 4 and 5 inches long, directly attached to the main stem, and can be highly lobed to relatively smooth. Plants will begin growing taller and get ready for flowering in midsummer. Typically the plants are only 2 to 3 foot tall, but I have seen plants as high as 6 feet in shady areas. Canada thistle stops actively growing when temperatures are above 85 degrees Fahrenheit, but can resume growth as temperatures drop in the fall. There is usually a flush of new rosettes in fall, but these will not flower and typically die off in the first hard freeze. The roots overwinter and will send up new shoots in spring. Colonies of Canada thistle are known to creep across the landscape and expand over time. Canada thistle is often considered one of the worst weeds globally. Canada thistle can spread in 2 ways, through seeding and also through their perennial root system. Creeping thistle can produce prolific amounts of seed, over 1,000 seeds per stem. The seeds are attached to fine white hairs and can be windblown. Seeds typically only establish in disturbed open sites, making Canada thistle a successful colonizer. Over 90% germination occurs within the first year, but seeds could be buried in soil and remain viable for a long time, up to 20 years. Canada thistle is more difficult to control because of its extensive, deep, creeping root system. This root system helps it form very large colonies and also stores a lot of energy that can help the plant recover from above ground disturbance. Vertically, the roots can be 6 to 15 feet deep, and lateral roots produce adventitious buds to form new above ground shoots. The lateral growth of a colony can be 10 to 12 feet per year. Densities of over 100 Canada thistle stems per square meter are not uncommon and can easily crowd out native or desirable species. When the roots are severed, like when hand pulling or tilling, root fragments as small as 1/4 of an inch or the size of a pea can form entirely new plants to start new colonies. New plants from root fragments can develop in as little as 2 weeks, and can be fully formed or able to flower and produce new adventitious shoots in around 8 weeks. If Canada thistle is tilled, infestations can become much worse. Tillage reduces competition for the thistle, and new colonies are formed through the fragmentation of the roots. This usually results in higher Canada thistle densities and can significantly reduce crop yields. It can be possible to decrease Canada thistle through tillage, but tillage needs to occur in 21-day intervals when Canada thistle is actively growing. This can be very time-consuming, expensive to perform, and create new soil disturbances that may provide an invasion potential for other weeds. Mowing can be an effective control if performed multiple times per growing season. Especially important could be mowing when temperatures are above 85 degrees as Canada thistle stops growing. Herbicide can be reasonably effective. Multiple applications are typically needed, and there is a chance of resistance forming because the large root system is so good at recovery. Herbicide may not be feasible due to the size of Canada thistle patches or the proximity to sensitive environments. If you have large, extensive patches of Canada thistle, and the goal is to reduce the density of thistle on a timescale of say 5 years with minimal input, reduce invasion potential for neighboring properties, and reduce disturbance of the land as much as possible, then biological control could be a good choice. Puccinia suaveolens is an adventitious fungal pathogen that can be found naturally occurring anywhere Canada thistle grows. It is typically introduced unknowingly with Canada thistle to new areas. It is highly host specific and will not infect any native thistles in North America. However, it is not a currently approved biological control through USDA APHIS PPQ. This is because it's a pathogen. Current wording of regulations means that pathogens for the control of weeds are considered biopesticides. Biopesticides are under the regulatory control of the EPA and require labeling. Getting a pesticide label involves a large investment of money and scientific study. It's highly unlikely a company would invest to do this work since the fungus is a poor candidate species for commercial production, and again, it's found naturally anywhere Canada thistle grows. This is still an ongoing regulatory issue that does not reflect on the safety or efficacy of the root fungus, but it does prevent the interstate movement of this pathogen and also prevents the commercial application of this pathogen. The information I will be providing today is for the identification and management of this root fungus within Canada thistle patches where it already occurs. As always, before considering collecting biological control agents, please ensure you have the proper permission or permitting to do so, and ensure the biological controls are permitted and allowed in your area. States and local municipalities may have other regulations. Puccinia suaveolens has been proposed as a biological control for Canada thistle since the 1860s. It is an obligate biotrophic root fungus. Obligate meaning it only survives in contact with Canada thistle tissue, and biotrophic meaning it feeds on living tissue. This is a fungus that lives inside of its host, Canada thistle. The life cycle of this rust may be divided in 2 stages, a vegetative stage and a spore-producing stage. The rust fungus is thought to remain in a vegetative stage inside the root system until conditions trigger the fungus to become reproductive and create spores. While within the roots, infection reduces root biomass through both root resources being used to produce defense compounds and Puccinia suaveolens directly feeding on these root resources. A reduction in the root resources of the plant reduces above ground growth, including flower or seed formation. A weaker root system can also make Canada thistle more susceptible to soil pathogens or reduce the plant's capability to regenerate from other control methods. When the rust fungus becomes reproductive, it will hijack the plant's growth and create systemically infected shoots in order to create spores. These systemically infected shoots are most common in early spring. They're spindly with larger spaces between the leaves, yellowish in color, and strongly floral scented. The yellow color is from both chlorosis, or the blocking of photosynthesis in the leaf tissue, and also the scented yellow-orange pustules known as pycnia. These pycnia produce pycniospores, which are the first of 5 spore stages. The pycniospores outcross and create acea, a structure that produces aciospores. Aciospores are chain-like formations of dark orange-brown spores. These spore formations will darken over time as the aciospores produce uridinia that will go on to produce uridinospores that give a characteristic rusty appearance to the plants and do not produce a floral smell. Systemically infected stems typically do not flower, die off early in the season, and may help to further reduce root resources of Canada thistle colonies. They are important to note in early spring, as the uridinospores produced by systemically infected stems will cause localized infections on neighboring plants. Uridinospores shed from the systemically infected stems onto neighboring plants and produce small localized infections that stay on the leaf tissue. Plants with localized infections do not have the same growth abnormalities associated with systemic infection. The small localized infections can continue to produce and shed spores, creating more localized infections throughout the summer. In fall, these localized infections can create telia that produce two-celled teleospores, the fifth of the 5 spore stages. Teleospores are the type of spore that can produce root infections and spread the pathogen systemically. The teleospore infected leaves fall off as they senesce heading into fall, and the teleospores either blow off and overwinter in the soil or germinate on new rosettes, where they can initiate new vegetative infection in the roots or systemic infection above ground. The localized infections should be checked with a good hand lens or jeweler's loupe to make sure it is rust fungus and not insect frass. A native lace bug, Porothuca distincta, is commonly found on Canada thistle and produces frass that can look similar to the fungal spores. Teleospores will be seen erupting out of the plant tissue, not just sitting on top. The teleospores will typically be more evenly distributed over the leaf tissue. Insect frass will look a bit dirty or sooty and is usually deposited in clusters or unevenly across the leaf. A jeweler's loupe, field microscope, or stereo microscope in a lab can be used to confirm if the blackish dots on a plant are frass or spores. Once you've identified spores and frass a few times with confirmation, you can usually distinguish the two with your naked eye. To collect and spread infection, teleospores are needed. This will require season-long monitoring and scouting. In spring, usually in May, patches of Canada thistle should be scouted for systemically infected stems producing pycnia. When systemically infected stems are found, you can use flagging tape, pin flags, or survey stakes with whiskers to mark these stems. These need to be marked since they will shed spores on neighboring plants but are going to die off early in the season. The markers will help you narrow in on where to find localized infections that produce the infectious teleospores in late summer. If there are a lot of these systemically infected stems, flags may not be necessary, as the entire patch might end up with localized infections. Periodically check back through the summer to make sure the plants are not being disturbed by grazing, mowing, or chemical application. The Canada thistle patch will need to be undisturbed. Beginning in late August to early September, Canada thistle plants with localized infections can be collected. To collect teleospore-bearing leaves, you will want gloves, long sleeves, hand clippers, and paper bags or large cardboard boxes. Check on the thistle patches you identified early in spring that had systemically infected stems. While collection typically happens in late August to early September, you want to time your collection for when the older plants are beginning to senesce but have not dropped their leaves. Begin looking at the plants adjacent to where systemically infected stems were present in spring. Confirm spore presence on the underside of leaves and cut the whole plant near the ground. It is possible to immediately place the whole stems in new Canada thistle patches without any further preparation to spread infection. I don't find this to be super reliable, but it has worked for me at a couple of sites in the past. To more reliably spread infection, you will want to prepare inoculum. This will allow you to create a consistent mixture of teleospores and plant material and quantify the amount being applied. For this, you will want to let the collected stems air dry for a couple of weeks. You don't want these stems to mold, so don't use plastic containers and check on them periodically. Once dry, the leaves can be stripped off the stem and then broken down into a fine powder. We use a kitchen blender to process this material, but hand crushing, a mortar and pestle, or another means of processing this material could also work. You will want this crushed material to be fairly fine, but there will be some clumpiness from the fibers of the plant material. This inoculum can then be stored in a cool, dark, and dry place until it is ready to be applied. To apply Rust Fungus inoculum, you will need the inoculum, gloves, and a way to lightly mist the plants with water, whether that is a handheld spray bottle, backpack sprayer, or hose with a misting nozzle. If you have it handy, a seed spreader can also be used. It's best to apply inoculum in the fall while Canada thistle has some active growth of rosettes. The best germination of teleospores occurs when temperatures are around 64 degrees Fahrenheit average, so 80-degree days and 50-degree nights. This most typically occurs around the first week of September in Colorado, but may be shifted a couple of weeks in either direction in any year or specific location. Locate the Canada thistle patch you would like to introduce infection. Applications are best done in the afternoon to evening, as the germinating spores can become light sensitive. Spray the Canada thistle lightly with water, and then you can place a pinch of inoculum per individual rosette, or if there is a carpet of thistle, you could broadcast the inoculum. You can use a seed spreader for this, but keep it low to the ground to avoid too much drift. Try to focus on getting inoculum in contact with living Canada thistle tissue. After applying inoculum, lightly spray the area again. This added moisture helps the spores to stick to the Canada thistle and also provides moisture for germination. When working to control perennial colony-forming weeds, it's always best to focus control efforts on the smaller satellite patches to eliminate the spread, and then focus on the perimeter of large established patches to reduce the population over time. However, if your patch is small, you could make inoculum applications throughout the entire patch. I like to take a photo of the weed patch, a GPS point, the collection site and amount of inoculum applied, and the date of release for my records. It could be possible to see systemic infection in the spring a few months after inoculations. On occasion, very large outbreaks of disease can be seen in Canada thistle patches. These large outbreaks are usually followed by a drastic decline in Canada thistle stem density and patch size. Sometimes full eradication of Canada thistle patches can occur. Even without large outbreaks of disease, gradual decline of the Canada thistle patch size and density should be seen. In Colorado, application of Rust Fungus inoculum in fall resulted in an average Canada thistle decline of 77% over 8 years. The rust fungus will not always produce above ground systemic infection. It can remain in the root system for years without sending up systemically infected stems. While systemically infected stems are a sure sign of infection, the absence of them does not prove an absence of infection within the roots. It's always a good idea to photograph, map, and measure weed infestation so gradual changes can be detected. Inoculations with the rust fungus can be performed multiple years in a row. The most compatible control method for an integrated weed management plan with rust fungus present is mowing. Summer mowing will encourage more localized infections and resulting teleospores in fall. Mowing in late fall, as the tall plants are senescing and rosettes are vigorously growing, can spread teleospores throughout the patch. In either case, mowing causes damages to the plants, allowing for more infection pathways and decreases the plant's defensive compounds, making the plants more susceptible to infection. Tillage and herbicide are not as compatible with rust fungus, but inoculum applied after other control methods may help control any new sprouts or invasion from Canada thistle. A healthy landscape with lots of competitive native vegetation may see better and faster results from control methodologies. Because of the extensive and regenerative root system, it will take persistent effort over several years to eradicate a Canada thistle infestation. I hope this video has given you the information, methods, and confidence to identify and manage Canada thistle rust fungus. There was a list of references and sources in the video description if you would like to read more about this interesting pathogen and how you can manage Canada thistle. Thank you for joining me today.